Hi, everyone. Thank you for joining today's webinar. I think we have quite a lot of people that have joined. Maybe we can start. Over to you, Dr. Vincent.
Just confirm if you are able to hear me.
Yes, we can hear you.
All right. Thank you very much, Nazi, and good afternoon to everyone. I take the opportunity to welcome you to our today's webinar, which is part of the 2025 African Pharmacogenomic Network webinar series. My name is Dr. Vincent Nyangwara. I'm a research scientist at the African Institute of Biomedical Science and Technology, and also Sydney Brenner Institute of Molecular Bioscience in South Africa. And I'm honored to serve as today's moderator. Just as you can see from the slide, our topic today is we want to explore the pharmacogenomic factors which influence the hydroxyurea, which is the commonly used drug for the treatment of the disease, and response in Tanzania sickle cell disease patient, which we are looking at it from the perspective of a genomic medicine approach. I take the opportunity to thank you all for joining us, especially our hosts, our collaborators, and also participants across Africa and beyond. Feel most welcome. And I will take the opportunity to introduce our speaker today. Our speaker is Dr. Siana Nkya. And Dr. Nkya is a senior lecturer in the Department of Biochemistry and Molecular Biology at Muimbi University of Health Sciences and Allied Sciences from Tanzania. In brief, it's called MUHAS. She's also the coordinator of the Office of Sponsored Projects. She's also serving as the head of MUHAS Genetic Laboratory and also the principal investigator and the technical lead of the sickle cell program in Tanzania. Dr. Nkya has over 15 years of experience in the field of molecular biology and human genetics, with her research focusing on disease prevention, diagnosis, genomic mechanisms, and also pharmacogenomics. She has played a leading role in advancing sickle cell research in Africa, including establishing one of the first genetic databases for sickle cell disease on the continent, and also co-leading Tanzania's newborn screening program. She has served in numerous and gotten a number of awards, which include the NIH Emerging Grant and also the AS Global Research Award. She currently serves as the president of the Tanzanian Human Genetics Organization, which is affiliated with African Society of Human Genetics. So we are delighted to have her with us today. And please join me in welcoming Dr. Siana Nkya to present to us. Thank you very much and welcome on board, Doc.
Thank you very much for the kind introduction. And I should start by really thanking the organizers for giving me this opportunity to share with you the work that we have done in Tanzania on pharmacogenetic factors that influence hydroxyurea response in patients with sickle cell disease. As you already said, which is very true, I'll just quickly pass through the background. I know probably most of the people in this platform are aware of what pharmacogenomics is, but I'll just quickly say that this is a field that we know focuses on looking at the influence of genetic variation on drug response by correlating gene expression or polymorphisms with a drug's efficacy or toxicity. What is the intention of pharmacogenomics? It's really to be able to identify, if possible, quite early on, individuals who are either likely or less likely to respond to a particular drug. And this information also help us to know or to at least determine patients who may require altered dose of certain drugs. I just wanted to say that when I was preparing this work, I actually found that pharmacogenetics is actually a not relatively new field. Some work was done since 1959. That was when we started to hear all the history of pharmacogenetic started, and people started to look at different candidate genes that can be influencing drug response. The benefits of pharmacogenomics. So we want to know if this drug is safe. We also want to use this information to select better drugs. We also can use this information for drug development or drug discovery. And ultimately, we want to be able to provide better patient care. So going to the study that we have conducted in Tanzania, we have looked at pharmacogenetic of hydroxyurea. Why hydroxyurea? This is because, as you said in the introduction, it's the only available therapy for sickle cell disease, which is known to increase levels of fetal hemoglobin. And fetal hemoglobin is known to be a major disease modifier for sickle cell disease. But what we have observed over years globally is that the response to hydroxyurea therapy varies from one patient to another. And it's now known that up to 30% of patients with sickle cell disease do not respond to hydroxyurea. And so it is important to know if there are any genetic factors that may influence hydroxyurea response or that we can use to predict hydroxyurea response. And of course, coming from Africa, we all know that Africa is highly genetic diverse. And so it's really important for us to be able to capture if any of these genetic diversity also inform in terms of hydroxyurea response. So although hydroxyurea has been linked to different, has been associated to different ways in which it can reduce the severity of sickle cell disease, we have focused on looking at fetal hemoglobin, and it's known that it does increase levels of fetal hemoglobin. And we know that when the levels of fetal hemoglobin are increased, we expect that we have decreased hemoglobin S, the abnormal hemoglobin in patients with sickle cell disease polymerization, which also decrease all the downstream complications of sickle cell disease. So the rationale of the study is that traditionally it takes months to determine whether or not a patient will respond to hydroxyurea. Muhimbili National Hospital, which is a national referral hospital in Tanzania, initiated hydroxyurea administration for sickle cell disease in 2016. And so we thought it's a great opportunity for us to utilize this platform to study, among other things, how genetic factors influence hydroxyurea response that we already also observe is variable among the patients that are seen at Muhimbili National Hospital. And so the proposed study was aimed at evaluating markers that may early on predict hydroxyurea response. Why? Because children who are likely to be poor responders, now, for example, in Tanzania, and I think in many places in Africa and globally, hydroxyurea is administered from 9 months of age. So you can imagine if you want to administer hydroxyurea at that age every day, it's a long-term administration of this drug. And if that child is a poor responder to hydroxyurea, they will be saved from unnecessary exposure to the hydroxyurea side effects, which are also known to be toxic. We can also use these findings to actually look at alternative drugs. And we know now that we have more and more drugs in the pipeline for sickle cell disease. And so we have an opportunity to actually consider alternative drugs or alternative therapies with more favorable side effect profiles, but also probably need less monitoring. Or think of combination therapy using hydroxyurea with another drug or with another intervention, rather than just using hydroxyurea, which may not be really effective for some of the patients. This was the conceptual framework of the study that we conducted, and we just looked at this, at hydroxyurea response at 3 different levels. The first level, which is what is traditionally used, is the hematological, looking at the hematological markers of hydroxyurea response. In this case, we focused on the levels of HbF, of fetal hemoglobin, F cells, but we also looked at distribution of HbF per F cells. We also know that as part of monitoring, we do full blood count and others. So in totality, hematological markers, this is one of the layers that we looked at or we categorized. So hematological markers of hydroxyurea response, but then we thought of molecular markers of hydroxyurea response. And in this, we looked at the differential expression of gamma globin gene over that of beta globin gene. Beta globin gene, we all know this is the gene that is responsible for synthesizing the beta hemoglobin. So in patients with sickle cell disease, we normally have a switch, or in every individual actually, we have a switch from the fetal hemoglobin to the adult hemoglobin. In normal people, we have the hemoglobin A, which is normal, but for patients with sickle cell disease, they would have the hemoglobin S. So we wanted to look at the response at molecular level, whether we are seeing more increase of gamma globin expression, which is the gene that is responsible for synthesis of fetal hemoglobin, because we already know that hydroxyurea is associated with increased levels of fetal hemoglobin versus the expression of beta globin gene. And then the third category is the genetic markers of hydroxyurea response. And in this case, we hypothesized that there would be markers in the gamma globin gene, which is directly associated with fetal hemoglobin synthesis. But because we know from the biology and from the genomic study of fetal hemoglobin synthesis, we also have other genetic variants that are not situated in the gamma globin gene, not even in the globin cluster. So 3 layers: genetic markers of hydroxyurea response. So we have genetic markers, then we have molecular markers and hematological markers. So the aims were to evaluate the pharmacogenomic response to hydroxyurea in sickle cell disease and to attempt to identify early predictive markers of hydroxyurea response. The primary endpoints that we measured were at hematological level, fetal hemoglobin levels, as I mentioned, at molecular level, the gene expression, and then at genetic level, we looked at different genetic variants. This included single nucleotide polymorphism, copy number variations, deletions, insertions, as well as gene expression levels on targeted regions. Secondary objectives were to look at the association of increased fetal hemoglobin or the distribution of fetal hemoglobin in red blood cells known as F cells following hydroxyurea treatment with clinical parameters, and also to compare hydroxyurea response and the sickle cell disease expression across different sickle cell disease haplotypes in Tanzania, Ghana, and Nigeria. So the study, the results that I'm presenting today are for Tanzania, but the study has been scaled up to also Ghana and Nigeria. So why did we go with this design? We looked at the supporting hypothesis. So the levels of the hydroxyurea response from literature is known to be influenced by 1, levels of fetal hemoglobin and individual possesses before the initiation of treatment. So the baseline fetal hemoglobin levels. But we also know that individuals' fetal hemoglobin levels is significantly influenced by genetic factors associated and non-associated with sickle cell disease haplotypes. It's not yet clear how baseline fetal hemoglobin levels influence hydroxyurea response. However, since fetal hemoglobin levels are genetically influenced, then we hypothesize that hydroxyurea response may have direct or indirect association with genetic variants that influence fetal hemoglobin. But we also know hydroxyurea as a drug may be influenced by genetic variants associated with the drug metabolism. However, we also don't know whether there's an interaction between genetic variants associated with fetal hemoglobin with those influencing drug metabolism. Why is this knowledge important? We wanted to use this knowledge to better understand hydroxyurea, to better inform on hydroxyurea administration and precision medicine, as I mentioned before, to be able to inform and influence treatment decision, such that patients can be administered alternative therapies or interventions. But also take into consideration mechanism of hydroxyurea response, especially when administering hydroxyurea, if in any case, in combination with other drugs. So this was the methodological approach. It was a longitudinal study. The total study participants are 450. This is from the 3 countries, Tanzania, Ghana, and Nigeria. The results that I'm showing here today are for Tanzania, which was the phase 1 of the study. We measured hematological parameters. They said gene expression profiles, and we also did targeted next generation sequencing. So now for Tanzania, we enrolled a total of 148 patients followed for 12 months. We categorized hydroxyurea responders in 2 groups, good responders and poor responders. And we did this by looking at the increased levels of fetal hemoglobin compared to the baseline fetal hemoglobin. As I mentioned, we focused looking at hydroxyurea response, in this case on levels of fetal hemoglobin. And so we categorized the good responders as those patients that had at least twice increased levels of fetal hemoglobin compared to the levels that they had at baseline before starting hydroxyurea. And the poor responders is those that the levels of fetal hemoglobin remained the same throughout the treatment. We measured the hematological profiles. We did full blood counts, high performance liquid chromatography to quantify fetal hemoglobin. And we also did flow cytometry to look at levels of the distribution of fetal hemoglobin in F cells. We did real-time PCR to look at the gene expression profiles, and we did targeted next generation sequencing using Illumina custom panel, which I will explain further a little bit later. And we targeted we selected known fetal hemoglobin associated loci, as well as loci that have been associated or are known to associate with drug metabolism. So the first step of the study, of course, after collection of the phenotype data, we designed the panel. And of course, as we all know, we considered pharmacodynamic genes, pharmacokinetic genes, just selected ones, and then modified, as I said, the ones that we know associate with fetal hemoglobin. So the composition of the hydroxyurea panel that we used looked at targeted loci that are known to influence fetal hemoglobin. This is what I'm showing on at the top. So including the gamma globin gene 1 and 2, BCL11A, which is a common locus associated with an increase with levels of fetal hemoglobin, HMIP, which is also known, and others. And we also selected a number of genetic variants that are known to associate to hydroxyurea metabolism. And we categorized the 2 groups. So although we started with 148 participants, but when we categorized the 2 groups in the way that I've explained, we ended up with 60 patients in one arm responders, and the other arm is low response to hydroxyurea. So this is just showing the target genes or loci that we looked at and how they are known and their role, or why the rationale as to why we included them. So we have a number of them that are associated with HbF synthesis, some of them enzymes that are known in pathways that are in the nitric oxide metabolism, some that are known to influence cell signaling, which is also a pathway. Both nitric oxide and cell signaling, these are pathways that are also influenced by hydroxyurea. And we also included two variants that are known, very well known, a family of cytochrome that is known for different drug metabolism. So we followed the normal steps of panel development, starting with defining the clinical use case. As I mentioned, the selection of which variants should go into the panel. And we also did an in silico design before going to the pilot wet lab. And we did this with support from the Illumina colleagues. And then we also tested the different performance targets, and we could actually see that using the MySeq platform, we could get the depth that we wanted, the uniformity that we wanted, and how to actually pull the samples that we wanted to use. Skip this, but then the strength of this design or the approach that we took is really to be able to look at multiple genes in a single assay and see how this information can actually help us to understand better hydroxyurea response and whether it can also inform in terms of dosage. The validation of the panel is ongoing. So we have a small support to validate the number of the genetic variants that we have looked at using Sanger sequencing. So the data was analyzed at different levels as we had categorized. So the linear regression was used to look at the association of hydroxyurea with hematological parameters. We did variant annotation and mutation prioritization using ANOVA. We looked at hydroxyurea-associated SNPs using Python. We did principal component analysis using agent software. We also did pathway enrichment analysis using GenMania and Enrich. And we also engaged other data sets, including the 1,000 Genomes Project Consortium data and African Genome Variation Project data. So at the hematological parameters, I'm just showing the results that we looked at the good responders, and you can see that we saw the increase of fetal hemoglobin from visit 1, from baseline up to the 3rd visit, which was around 3rd month. We saw increase in fetal hemoglobin, but we also saw increase in mean cell volume. And this is probably because of the increase of F cells, which are known to have larger volume compared to the red blood cells, to the other red blood cells. We also saw a significant reduction in white blood counts and reticulocytes, which the two markers are known as markers of severity, hematological markers of severity in sickle cell disease. For gene expression, we also observed differential gene expression of the gamma globin. So I'm showing, if you can just focus on the selected red box, we are looking at the differential gene expression of threshold of gamma globin gene compared to the beta globin gene. If you look at the first visit, going to the 3rd visit, you could see that there's an increase of gamma globin gene. So the point here, why we did this, is because we are also trying to establish whether we could use the molecular markers as markers of early prediction of hydroxyurea response. And here we see that you can start to capture it much better from maybe 2 months and ahead. And then for the genetic, the next generation sequencing results, we looked at different aspects, and here I'm showing the frequency of different variants in the targeted genes, and the red line represents the non-responders or the poor responders. The black line shows the good responders, and generally the frequency, or we had higher frequency of variants in the responders compared to the non-responders. And we interpreted this as probably, as we all know, that there could be cumulative effects from the different single nucleotide polymorphisms in these targeted genes or loci that influence hydroxyurea response. And the same thing was also observed in the general population. As I said, we also engaged other databases, and we could see from the general population, African general population, that there was also a varying frequency in these SNPs, in these targeted loci, confirming what we observed. And so we think that the different SNPs, the different effects from the different SNPs may influence different levels of hydroxyurea response. Of course, we had some distinct differences, for example, for this CYP to C9, which was quite different. And on the right, we also looked at the pathogenic single nucleotide polymorphisms in the targeted genes. And here we saw that, again, the red line represents the non-responder and the black line represents the good responders. And we saw a high frequency or high percentage of pathogenic single nucleotide polymorphisms in the non-responders compared to the good responders. So we had low rare variants and low frequency of pathogenic SNPs in the good responders. When we looked at the principal component analysis, we saw that in the general, so in the top box shows the different groups in the African population, and the bottom one shows the different components using our data sets. And you can see that at the top, we could see variable genetic, but very distinct genetic pattern that may influence hydroxyurea response. And at the bottom, we could see that for the good responders, so the green X is the responders, and then the red triangle is for the poor responders. And we could see that for the good responders, we had variable pattern, while the pattern for the non-responder or poor responder was more homogeneous. So again, we think that we assume, or we interpret this as, again, having different SNPs, genetic variants that have different effects that influence the variable hydroxyurea response. And when we looked at the pathway enrichment, we had different interactions or different enriched pathways, including physical interactions, looking at also genes that are co-expressed, co-localized, genetic interactions, shared protein domains. And most of the pathways were around the biological functions, including tetraviral binding, oxido reductase activity, antioxidant activity, hydrogen peroxide metabolic process, drug catabolic process, drug metabolic process, and response to drug in general. But if you look at this figure, you will see two distinct patterns that for us were of interest. The first one was with enriched pathways that associate with loci that are known to influence fetal hemoglobin synthesis. So You can see HBG1, HBG2, and other linked to other loci. But we also saw that the drug metabolism pathway, including the being linked to the 2 targets that we also sequenced. And we think that these results are really important, and we need to actually fine map, because using these results we could actually identify other loci or other variants that may also influence hydroxyurea, but may also be potential for other drugs that may work as how hydroxyurea work, but with probably less effects. So in just a few remarks for conclusion, in contrast to poor responders, the good responders tended to have a lower proportion of pathogenic SNPs, as I showed. These findings suggest that potential cumulative mutations at these genes might be beneficial for good response. The different SNPs, as we saw, high frequency of SNPs in the different variants, which was much higher for good responders than the poor responders. So they may have cumulative mutations that are beneficial. And then enriched, we have observed 2 major pathways: enriched fetal hemoglobin synthesis pathway and drug metabolism pathway. So we have had 2 publications out of this work to date. The first one describes how we developed the panel and the details, and the other one describing the results, the pharmacogenetic results, as well as the other the hematological ones. How are we thinking of applying this knowledge? We are hoping to conduct more studies. As I say, the validation of the panel is ongoing. We have also received support to scale up this study to Ghana and Nigeria. So we are hoping that we'll have more results and we'll be able to validate some of the things that we have seen, but also probably discover other findings that we did not, due to limited sample that we had. But ultimately, we are hoping that we can use this knowledge to inform in a pharmacogenomics panel for hydroxyurea, which I think will be a capacity that we can use even for other drugs in Africa, for sickle cell disease, to enhance precision medicine. We also are keen to use the results that we have found for drug discovery. So really identifying other potential variants that can be targeted for treatment for sickle cell disease. I'm just showing this because I saw this, and this is just a trend of the global pharmacogenomics market, which is going very high and is expected to continue to be high. But on the right, you can see that Africa is really lagging behind in terms of pushing for pharmacogenomics and its application in terms of precision medicine, but also in terms of informing and contributing to the pharmacogenomics market. We all know that in Africa, we still we need to work around the barriers that may influence the applications of pharmacogenomics. So we still struggle with complexity of finding gene variations that affect drug response. We find millions of SNPs that need to be analyzed and really determine their involvement in drug response. Many genes are likely to influence response in different ways. We have limited knowledge of which genes are involved with each drug response because the drug responses could be variable. We still have to establish more frameworks around confidentiality, privacy, and the use and storage of genetic information for pharmacogenomics studies. We need to continuously educate healthcare providers and patients and families on the uses of pharmacogenomics, which if we don't do this appropriately, complicates the process of prescribing and dispensing drugs. Physicians must execute an extra diagnostic step, which is not a tradition in many African settings. Therefore, we really call for better understanding of genetics by all physicians across the continent. For companies are usually not interested to make multiple pharmacogenomic products because it's not beneficial. There's no profit in that. Most pharmaceutical companies have been successful with their one-size-fits-all approach to drug development. And for small market pharmaceutical companies, have to spend hundreds of millions of dollars on pharmacogenomics-based drug development. So with that, I would like to thank different people who have really contributed to the work that we are doing, the work that we did. So first, patients and families. We received funding for the results that I've presented from the American Society of Hematology, and we have received support from Novartis Global Health Foundation to scale up the study. The sickle cell program of Tanzania, the research team that have really worked very, very hard for this work. Thank you.
Thank you very much, Dr. Siana, for that insightful presentation. We'll now open the floor for questions. So please, you can post your questions in the chat box, or you can raise your hand in the function bar if you'd like to speak. But first, I will start with the question which has been posted on the chat box. I will just read them out so that Dr. Siana can respond to them. So I will start with the first question from Adrian. So she was asking, to what extent does the fact that in some low and middle income countries, hydroxyurea is prescribed at a fixed dose rather than in the recommended weight-based dosing factor in the 30% of poor responders in this case? So she is asking, is the 30% finding for those with perfect adherence?
Thank you very much. So yes, so adherence, that's an important point. And I wouldn't say that it was easy to follow the patients. So the approach that we used, because we could not really measure drug levels, which would be the most appropriate way of measuring compliance and to ensure that patients are really taking hydroxyurea as prescribed. We did follow-up every week, using phone call to remind patients to take the drug, but also to ask them like probing questions, just roughly, because we would know that when they last visit the health facility, they were given this amount of hydroxyurea. And if you ask them and then see that this is not as what is expected, then you would know that probably something might be wrong. That was one way that we did monitor adherence and compliance to taking hydroxyurea. But we also, because we were doing full blood count, we also were observing very closely the full blood count parameters. And if in any case we observed that there were abnormalities, we would go back to check and recheck with the patient and ensuring that they were actually following and complying to the drug prescribed. And whenever we saw that they are not complying, we did excluded them from the analysis. Thank you.
Thank you very much. Just a follow-up question. Adra also was inquiring, for the poor responders, was ineffective erythropoiesis a potential factor, and are there any of the mutation in the tested panel associated with the IE phenomenon? He cited a literature down there, but I think his question was more for the poor responders. i.e. a potential factor, and are there any mutation in the tested panel which have been associated with it?
So yes, so I agree with Adrian that there's levels of fetal hemoglobin have been associated with ineffective erythropoiesis. And I think from the literature, from the different literatures, we see that ineffective erythropoiesis leads to the synthesis of F cells, which are the cells that carry fetal hemoglobin. So I would say that based on that, yes, hydroxyurea response would be influenced by ineffective erythropoiesis, because we already know that hydroxyurea response is influenced by the baseline levels of fetal hemoglobin that an individual has at the time, even before hydroxyurea administration. So some of the genes that we have included in this panel are those that are associated with fetal hemoglobin. I can't say that they are directly associated with ineffective erythropoiesis, but they are associated with fetal hemoglobin. So for example, MIB is known to influence hematopoiesis. So yes, it could also influence ineffective erythropoiesis, but we have to look at this in more detail. Thank you.
Thank you very much, Dr. Siana. Risi Wesonga is asking, she's thankful for the presentation, but she was asking, were there any anomalies in terms of fetal hemoglobin levels whilst grouping the patient in the two categories for the responders and non-responders? And did you have any cutoff for the baseline in your methodology?
Okay. So we didn't have a cutoff for the baseline, but we did have a cutoff to categorize whether a patient would fit into a poor responder or a good responder. And the way we did this was to look at the baseline levels. So this was individual based. So we looked at the levels of fetal hemoglobin at baseline before hydroxyurea treatment. And then after 3 months, we looked at how the levels were. And if they had doubled or more than doubled, so our threshold was for someone to be a good responder, they would have twice as much HbF levels as what they had at baseline. And for poor responders, it will be the same or roughly the same. That's how we categorized the two groups. Thank you.
Thank you very much, Dr. Sian. Prof. Colette Dandara is asking, in different studies, they're generating evidence to show the role of pharmacogenes. So what do we need to overcome the implementation barrier you just highlighted in your slide? Because currently there seem to be no pharmacogenomic application in African patients.
Yeah. Yeah, so I share Professor Colette's concerns. I think that we need to put more. One, I believe with all my heart that we need to push pharmacogenomics in Africa. Why? Because we have had enough examples that have shown that we receive drugs from elsewhere and they are not, one, they might not be effective for our populations, or they might be even lethal for our populations. So I think we really need to push for pharmacogenomics, pharmacogenetics in Africa, because we have very high diversity and we cannot just go with an assumption that any drug will just fit all the categories of the patients that we have, given the high genetic diversity. So I think we should push for pharmacogenomics, pharmacogenetics. But as the normal trends we have seen, even for general genomics, investment, research, yes, we see more and more investment now, and we are hoping that through that we can also put more investment for pharmacogenomics as a very direct application of genomics in Africa. And so I think we should argue for more investment in conducting pharmacogenomics research in Africa, but we may also advocate to ensure that as we push genomics agenda in Africa, we should leverage the different resources that are being poured to conduct genomics research in Africa to also inform on pharmacogenomics. I think there's a way that we can do that. We can leverage the genomic study to also include some of these aspects, some of the genes that are known to influence drug metabolism in the different aspects of the genomic study that we do. And I think the last thing that we can easily do is really starting with what is known. So for example, there are different panels that are known for cancer, for different diseases that are known. And we know from biology, some of the genes, for example, the cytochrome family is known for influencing drug metabolism. So we can start from what is known and doing simple studies to show more and more that we actually need to conduct pharmacogenetics research and ultimately actually having pharmacogenomics supporting medicine in Africa. I'm not sure if I have answered the question, but I think those are my thoughts. Thank you.
Thank you very much. I think Prof. Colette has something to say. He's raising her. Maybe we can allow him and then we continue with the questions after he has addressed his concern. Welcome,
Thank you
Prof.
very much. Very good, interesting presentation. And yes, you responded very well. Do you think it's time that we should put a pharmacogenomics implementation working group for Africa? Because I think, like you say, we're going around in circles. We're picking this and like this data, we'll leave it, and then we'll start working on another piece of study and show evidence and move on. If you look at the CPIC, they start to put on working groups that really focus on evidence, and then they build, and then so that we start from somewhere and we can continuously improve whatever tests we come up with. What's your ideas on that? Thank you.
Thank you so much, Professor Colette. I could not agree with you more. I think that would really help in terms of streamlining the resources that we have to, as you're saying, and that is always our challenge, doing things in patches, and then we can't actually pull the evidences together to have one voice that shows the importance of what we are doing. So I think having that technical working group in Africa would really help to streamline a number of things. So the resources also putting the agenda, what is important for us, what in terms of pharmacogenomics, what do we want to address? There are a number of things that are there to be addressed. So one is research, but then there's training, there's awareness, there's also saying which diseases should we prioritize in terms of applications of pharmacogenomics. So I think having a technical working group would really help to streamline a lot of things. Yeah.
Thank you very much, Dr. Siana. I hope Prof. Colette's concerns have been addressed. I believe so. So just to echo what you have said, I completely concur with you with respect to diversity in Africa. And what I think is collaboration will be very key for us to be able to ensure that Africa as a continent is well captured. If you look at South Africa, maybe collaboration from South Africa, Eastern Africa, Western Africa, Northern Africa, so that we can have kind of a reference database. Though it may not be homogeneous, but it can give a reflection on how diverse our continent is when we want to implement pharmacogenomics. And I think that is what ABIS is trying to do, where they're having all this kind of research in better part of Africa so that they can have like a consortium where we can have a reference kind of database. And maybe I can go to the next question. Did you experience any challenges in designing the panel? Maybe we can learn from that. I think that is what Marilyn is asking.
Yeah, we did. It was not straightforward. And I forgot to say that we did this work in Tanzania. So the sequencing was actually done in Tanzania at Muhimbili, Hematology Clinical Research Laboratory. And we were working very closely with the Illumina team when we were designing the panel. And you can imagine, we have just looked at a few variants. Yeah. So even the selection of which locus we should focus on was not very easy. Of course, we used the knowledge that we had acquired from our genomic study in our population, but also others that have been reported elsewhere. So we did research a little bit to actually prioritize which ones we should go with. If it was for our wish, probably we would have done whole genome sequencing or something like that, or whatever, but because we wanted to explore. But because we had to work with what we have, we had to really strategize on which ones and how many should we put in. So we did receive a lot of support. So it was about which ones should we put into the panel, and then the designing itself, making sure that we have the right chemistry to actually capture the data across these variants in a way that is still going to be quality data and we can analyze. So we had several iterations of which combinations could go together. And also, of course, we did the pilot with the Illumina team to see if actually that panel would work before we actually we employed it for the study. So yes, there were challenges, and I think that this is an area that we continuously need to find ways of doing better, the designing of the panel, so that it's actually informative in the way that we want. For us, we could get some support from Illumina in doing this. Thank you.
Thank you very much. I just had a question. I may not get time to ask it based on just the technical problem associated with the making of the panel. So from the finding you've just shown us, what genetic factors would appear to be having the strongest predictor of hydroxyurea in these patients?
Okay, so I'll go back to, so that's a good question. And that's why we mentioned that. So we had a number of significant variation in particularly in the genes that associated with fetal hemoglobin, but also in those. So you can see, for example, I'm showing these results, and if you look at the frequency of SNPs in the different genetic variants, you would see that we had a significant difference in HBG1, right? And also in SARIA1, in MIB, okay? And then also in KLF10, and a little bit for NOS1. We had significant high frequency of SNPs in CYP2C9. Although if you look at the pathogenic variants, we also had a number of pathogenic variants in the same locus in the non-responders. So we could not really make a decision about that one. And if you look at the pathogenic, the frequency of pathogenic SNPs in the different genes, again, you will see that we had a significant difference in MIB, which is in chromosome 6, and it's a known modifier of fetal hemoglobin levels. A little bit on HBB, CYP2E1, in HBG2, in AG92. And so what we are doing now before we say for sure that this could actually be used for predicting hydroxyurea in a clinical setting, we have to validate this. And I mentioned that we have an ongoing study to do a validation, but we also are hoping to have more results as we have expanded to have more samples in Tanzania, but also in Ghana and Nigeria. Yeah.
Oh, all right. Thank you very much. I will combine two questions from George and Risi. George was asking, he was wondering if nutritional status was factored in as a confounder when categorizing non-responders versus responders. And then maybe Risi was asking, based on your findings, what recommendation did you give to the on-call clinician for patients who were non-responders to the drug? And how did your result directly influence the uptake of the medication in your study site? Thank you.
Thank you very much. These are all great questions. So no, we did not include nutrition as a cofactor in this study. It will be interesting to look at that. I think I have had colleagues telling me to look at also microbiome. So this is something that we can look at because microbiome may influence metabolism of drugs, so even hydroxyurea. So this was for the other questions. So I would say that this is a translational study. Hopefully we will be able to actually translate these findings into clinical application. This is what we want to do. This is what we are keen to do. But at this stage, we are just using this study to start to sensitize the health community on the role of pharmacogenomics in medicine, which I think is very critical. Although we are not yet at a point where we can tell the doctors that now before you prescribe hydroxyurea, we need to sequence this particular gene and predict and tell you for 100% that no, this person would not respond to hydroxyurea, but that's where we are heading. At the moment, we are just using these findings to sensitize, to start to show that there's something, because we know that we see variable hydroxyurea response in our sickle cell disease patients. And although we know that there could be environmental factors, including nutrition and others, but we know for sure that there also could be genetic factors. And we know this because there are patients that go to maximum dose and they still don't respond to hydroxyurea. And we know from biology that if you cannot metabolize a drug, for example, you can't metabolize the drug. So even if you pump in a lot of it, doesn't make a difference. So, and we know metabolism is influenced by different enzymes in different pathways, which is informed from the genetic variants that we have. And that's why we also included some of the enzymes that we know are included in the pathways associated to hydroxyurea. So yes, we are not yet there to go to the clinic, but this is just the beginning of that journey. Thank you.
Thank you very much, Doc. I see we are almost past time, but I just had one general question, not necessarily on what you have presented, but I was wondering, how can we ensure that advances in pharmacogenomics, especially in Africa, reach underrepresented population and not just those in the research hubs?
Sorry, say it again.
Okay, just like a general question, not necessarily on what we have talked about, but I was wondering, how can we ensure that advances in pharmacogenomics and genomic medicine reaches underserved population and not just in those areas which we have the research hub?
Okay, that's a great question. So I think one of the advantage that we have, which I've not mentioned, in Africa now, I think we are really at a point where we can leapfrog the advancements in genomics for two reasons. One, we can see that technology advancement and the cost is going down. So we have great technological advancement and the cost for that technology is going down by the day. So we can actually tap into that. Number two, we now have more tools. We have free access to, different tools that we can use. Anyone with a phone somewhere, they can access something. So we should also leverage on that. We have artificial intelligence, machine learning, modeling, and all that we can access now that we could not back then. And I think Africa should really make sure that we leverage on these two advantages. The technology cost going down, and then the access to different tools that we can use to analyze our data and improve and improve. And I think if we are able to do this, this means that we will also develop panels that are, at a cost that can be accessible to the very lower levels. And that's what we are hoping for. Thank you.
Thank you very much. Maybe the last question will come from Risi Wesonga as we now wind it down.
All right, thank you for the opportunity and thank you for the presentation, Doc. This one is purely out of curiosity. Given that we have some patients who naturally have high levels of fetal hemoglobin, and hydroxyurea tends to also increase the fetal hemoglobin levels to help patients, sickle cell patients, in terms of improving the crisis rate. I just wanted to find out if maybe from the additional studies that you're doing, is that something you'll be looking into or you're already looking into as to whether does hydroxyurea have any impact on these individuals, or do they need to take it or not need to take it, or if they're taking it at what quantity, something like that, just out of curiosity.
Okay, so again, it's a great question. We did look at it to a certain extent because we based our design on the increased levels of fetal hemoglobin. So we know that when we talk about increase in fetal hemoglobin, it's not just the increase, but also the distribution of the fetal hemoglobin in the red blood cells, in F cells, to a considerable amount that will actually inhibit hemoglobin S polymerization. So at the moment, at least from our population, the increase that we observe for fetal hemoglobin has not passed that threshold where distributed in F cells to an extent where someone may not need hydroxyurea. But I think that's a great question, meaning that even when we are thinking of the dose for hydroxyurea, because it's a drug that is being used long-term, we need to take that into account. But I think more necessarily is because we are interested at the genetic variants that influence levels of fetal hemoglobin. I think we need to pay attention that the individuals that already have genetic variants that will influence fetal hemoglobin might need a different dose because already they are making fetal hemoglobin at a level that is different from the others. And because they already have this natural ability, they may not really require the same dose as one who cannot naturally synthesize increased levels of fetal hemoglobin. So I think I agree with you that this is something that we need to take into account. Thank you.
Thank you very much, and I hope Racy's question has been exhaustively answered. And as we now wind it down, I would like to thank the African Pharmacogenomic Network and also appreciate Dr. Sian for the excellent presentation and all of you for your active participation. And just a recap that today's session has highlighted the importance of pharmacogenomics in ways on how we can improve treatment outcome for patients who are having sickle cell in African continents. I just want to encourage you to stay engaged with our network and join our future webinars. Mine is to wish you well and have a wonderful day ahead. Thank you very much.
Thank you very much. Thanks, everyone. Bye-bye.
Bye. Bye, everyone.
Bye.
Bye.
I've never